Hi, my name is Hiroshi Tokuo. I'm a research assistant professor in the Department of Physiology and Biophysics at Boston University School of Medicine. Although previously I was a practicing dermatologist, currently I'm a full-time researcher interested in the molecular mechanism of melanoma metastasis. I'd like to thank Outrun for funding my research and for giving me this opportunity to share with you my melanoma research. As many of you have noticed, black cats and dogs frequently have white feet and white belly spots. These black colors are produced by melanocytes, pigment cells responsible for the coloring of our hair and skin. White belly spots are caused by a delay in melanoblast migration during embryogenesis. Melanoblasts are precursors of melanocytes and migrate long distances during body development as shown by arrows in this picture. The feet and belly are the farthest destination for this migration, so a delay causes blank area of melanocytes. We found that mice that do not express the motor protein myosin 10 have white belly spots, evidence that myosin 10 is necessary for melanocyte migration. But why are white belly spots important for melanoma research? Melanoma frequently metastasizes, that is, the cancer cells spread from the place where they first formed to other parts of the body. This is a major cause of death in melanoma patients. Our general concept is that melanoma metastasis uses the same mechanism as melanoblast migration in embryos. Thus, we speculate that our gene of interest, myosin 10, controls not only melanoblast migration but also melanoma metastasis. I'll introduce a little bit about myosin 10. This is the molecular structure of myosin 10. Myosin 10 interacts with actin filament with its motor domain, which converts the chemical energy of ATP to mechanical work, resulting in its movement along actin filaments by a hand-over-hand mechanism. And the tail domain binds to specific molecules. Thus, myosin 10 functions as a transporter in cells. Using actin filaments as tracks, it transports specific cargo molecules to specific intracellular sites. So what happens in melanoblast cells after myosin 10 depletion? This is a representative image of cultured melanoblast cells. Many control cells show long pseudopod-like protrusions. In myosin 10 depleted cells, long protrusions are reduced, and migration speed is slower than that of control cells. Then the next question is, what happens if myosin 10 is depleted in melanoma-inducible mice? As we speculated, we found that myosin 10 depletion in melanoma-inducible mice significantly decreased melanoma metastasis. Thus, myosin 10 plays important roles not only in melanoblast migration but also in melanoma metastasis. Surprisingly, myosin 10 depletion also delayed the onset and development of melanoma. We also found that myosin 10 is highly expressed in human melanoma samples, and increased expression of myosin 10 decreased patient survival. However, little is known why and how myosin 10 depletion inhibits melanoma development and metastasis. The Outrun Scholar Award will give us an opportunity to investigate this mechanism in depth. For example, what cargoes of myosin 10 are responsible for melanoma progression, and how does myosin 10 work in other cell types such as immune, stroma, endothelial cells for melanoma progression. By accomplishing this project, we hope to gain insight into how to prevent melanoma progression, resulting in the development of new types of drugs for melanoma treatment. Thank you.